Good morning, everyone, and welcome to Breakfast at UMHealth 2024, episode 36. I'm Thibashini Nair Sathasivan, moderator for today's session. It gives me great pleasure to introduce our speaker for today, Dr. Krishnamurithy Genasan. Dr. Krishnamurthy is a senior lecturer in the Department of Physiology, Faculty of Medicine, University Malaya. He was awarded the prestigious University Malaya Bright Sparks and dual PhD JPA scholarships, completing his dual PhD in orthopedics biology from University of Liverpool and University Malaya in 2019. His research focus includes osteoarthritis-associated immunology and regenerative medicine. Recognized for strong leadership skills, he was recently appointed as the head of Central Research Laboratories. Today, Dr. Krishna will be sharing insights on the research facilities and services available at the CRL with his topic title Central Research Laboratories, a Catalyst for Innovation and Growth. If you have any questions for Dr. Krishna, please feel free to enter your questions in the Q&A session. Over to you, Dr. Krishna.
Thank you, Tiba. Thank you for a nice introduction. Hi, everyone. I hope everyone having your tea or coffee next to you while watching this presentation. So basically, I'm not going to talk about osteoarthritis or inflammations and so on and so forth. But basically, I'm just going to share some information about the facilities under the operations of Central Research Laboratories. All right. So basically, I believe everyone knows that where the Central Research Lab. So we're going to share you what are the different subunit under the Central Research Lab and what are the facilities or resources that everyone can use to the best for your research progress. All right, so I'm just going to share a short video. So while we are running the video, then I will give you a bit more details about the facilities along the way. So let me share the video now. All right, so as you guys can see here, so under the Central Research Laboratories, so basically we have five subunits laboratories. So the one including a medical biotechnology laboratory, then Cell and Molecular Biology Laboratory, CMBL, Transnational Core Laboratory, TCL, and we have the FOM calibration unit, and as well as we have FOM supply store. All right, so this is our what do you call our CRL team. So basically, we have two senior science officers, Ms. Tiba and Ms. Melissa, and we have about 12 support staff. So each and everyone will introduce themselves during the video running. All right, so this video just to highlight, to show you the locations of all the laboratories. So I believe you guys know where is the quadruple angle of the faculty right opposite the open space of the Faculty of Medicine. And then we have CMBL. CMBL is in a different building, totally in different building. Basically, you can see the glass door on the way to nostril entrance. You can see the glass door on the right-hand side. You can see the signboard for the CMBL. The Translational Core Laboratory, the TCL, is a bit distance from Faculty of Medicine. It's under the one of the buildings in University Malaya Medical Center, which is the 11th floor, the top floor of the KWKK. And then we have supply store. I believe you guys know the supply store in our open space. There is a big signboard there. And also the FOMCAL, the FOM calibration unit is available within the medical biotechnology lab.
Hi, good morning. Please register your name here. Thank you.
Hi, welcome to Medical Biotechnology Laboratory. At MBL, we house both proteomics and genomics facilities along with our FOM calibration lab. Now, let's take a closer look at the proteomics facility.
All right, so now we are entering the first subunit within the medical biotechnology lab, the proteomics unit. So the proteomics unit basically equipped with A to Z if you want to do any sort of a protein analysis. As as simple as the protein quantification, like Bradford assay, until the protein identification using LCMS.
I'm doing SDS page. SDS page is vital for various applications in basic or clinical research, such as protein identification. And we use that protein for further analysis like Western blotting. Let's analyze.
All right, so gel electrophoresis is one of the prerequisites and basic what they call the parameters in protein analysis. So basically, the proteins that we extracted from the clinical samples, basic science culture samples, we use the gel electrophoresis to separate the proteins into the bands according to their weight, and then we use the specific band or the protein that's separated for the further analysis.
After the gel is prepared, we use this gel dot system to analyze the gel image. Simply place your gel inside this instrument and use the software. This software helps us to view the image and capture the protein band for future analysis.
All right. So basically, these gel dot machines that you are seeing here will be replaced with the brand new, most advanced gel dot. So the most weighted, the machines will be arriving very, very soon. So it is under the procurement basically. So Puan is one of the our what do you call the senior staffs under the proteomics. She's her expertise is really excellent. In fact, I would say she really helped me even for my PhD study many years back to identify protein samples from my tissue samples. And this gel dog will comes with its software where it gives very clear the band images that you can see here.
I am performing in-gel and in-solution digestion of proteins to break them down, which will help us identify our protein of interest using mass spectrometry. It is essential to perform thorough protein digestion to ensure reliable results.
So as Puan Atira mentioned just now, so basically the bands that we identify will be separated into an individual the bands, and then we digest the band like we call in-gel digestion or in solution digestion to get the protein into a simplest form before we run through using high-end machines like LCMS.
this digested protein is ready to be analyzed. If you like to know what type of protein and protein identification of this unknown sample, we use this liquid chromatography mass spectrometry or LCMS. LCMS is very powerful tools to do a protein profiling and other proteomics application. In proteomic facility, we provide very comprehensive service analysis, including sample prep, STS page, protein digestion, and data analysis. If you would like to know more, kindly email us at proteomics@um.edu.my. So let's explore more on our other service.
All right. So when it comes to the protein identification using LCMS, QTOF, so this machine is high throughput machine. Is advanced machines to separate your protein, identify your protein using UniProt, and then we can use the PIC software. Or now I think we're going to install a new software. I think it's a more advanced software to tell you the protein of interest that you are looking for, and then the further protein that identify and the protein of interest that listed down will be used for further analysis like validation using Western blot. Ponsri and the team will help you, in fact, and give a consultation. And I just want to highlight that the proteomic unit has its own optimized protocol from starting from protein extraction until protein separation and identification. So please do come and approach us and talk to us. So then Ponsri and the team can give basically a free consultation for our faculty members, especially the young researchers, but not limited, can be the seasoned researchers as well. And come and approach us, we can give you a free consultation. And then, of course, there is some minimal charges. You may ask why we still charge, because each and every machine, especially these high-end machines, requires maintenance. So the Faculty of Medicines basically maintain this machine using the charges that we impose for the service. All right. So and that charges as minimal, and we have a package or combo package where you can have a number of acquisition and etc. Then the charges will be even more affordable.
Next up, let's explore the genomics facility.
All right, so when come to genomic facility, so it's another subunit under the MBL itself. So in genomics facility, so basically we have A to Z, what do you call the service. We can advise you in terms of how to extract the genetic component, either DNA or mRNA, and then how to measure the concentration, and then how to run that mRNA for the using the real-time PCR. And in fact, we have a multimode plate reader. The multimode plate reader has various function in terms of its fluorescent measurement. Let me see, I need to recall the details. The fluorescent resonant energy transfer, the FRED, also in the fluorescent polarization. So all these different, what they call the techniques is already incorporated in this high-hand multimode plate reader. And in fact, the charges is basically, as I said, a very basic charges for the maintenance of this machine, but still affordable for our faculty members.
Hi, let me introduce you to our BD-FAX Canter 2 flow cytometry. This is a powerful tool equipped with the two lasers, the red and blue. It offers a lot of cell characterization and also is a high-speed cell analysis. It's not enough for you. Let me show you. We have another one of a flow cytometry family member in our.
All right, so let me introduce our flow cytometry equipment. So this machine basically looks like the machine itself looks like quite old, but basically all the parts inside is a brand new. So we already replaced with new lasers, and we already calibrated the machines for the top function. And this machine comes with the two lasers, blue and red lasers. It's really helpful for some basic analysis, cell phenotyping, um, cell viability analysis. So this is very helpful for those who want to identify different types of cells based on the surface marker expression. So please use this machine. And in fact, we can give a free consultation, and we are in the pipeline to prepare free consultations in terms of how to design the study for the customer. Depends on the the aim of the project. And then we also organize a what they call flow cytometry workshop in collaboration with industrial partner to teach you, to educate you A to Z how to operate this machine, how to troubleshoot this machine, although we will help to troubleshoot this machine, and then how to design your study for the facts analysis. So now Puan Nadia will take you to another level, which is the high-end machine in flow cytometry.
Facilities. Over here, we have the BD-FAX Area 3. This is a more powerful and advanced machines that takes flow cytometry further by offering cell isolation and cell sorting. Okay.
All right. So as you can see in this picture now, so this is a FAX area. At the moment, it's only functioned with two lasers. We're trying to replace the third laser, depends on the demand that we have. So many of you may not aware of the the capability of this machine. So these machines not only can be used to phenotype or identifying your cell of interest. However, this machine will help you to separate, to sort your cell of interest in an aseptic environment or condition. So these cells identified in the aseptic condition can be used for the further cell culture activity. So this will help you to work on a specific cell type from a pool of cell that you isolated from a tissue sample, tumor samples or cancer tissue samples, any sort of a tissue that can be, the cells can be isolated and can be run through these machines to identify and separate the cell of interest for the further analysis. Although the cost is a bit pricey, we try to revise the price, make sure that it is affordable for our faculty members and also for the outside users. However, please come and talk to us, and we can give you a free consultation, and we can discuss about the package that are available for you. Our main aim is to make sure that this machine can be used as one of the parameter in your research objective, testing. So this, can produce very good papers. I would say even myself, I use these fax machines to publish so many papers. Even one of the papers that I published recently in Frontiers in Medicine. So please utilize this equipment to your best and support your research aim. Thank you.
But if you're looking into molecular studies, I will pass you over to my colleague that we have offered the qPCR.
use our Step 1 Plus real-time PCR system. It offers accurate quantification of the PCR products in real time. With its easy-to-use data analysis and user-friendly software, it is perfect for your research in gene expression, genotyping, and viral load analysis, and many more. Do you know, to achieve optimal...
All right. So before move on to further explanation, so I just want to introduce. So we have 4 unit of real-time PCR machine. So 3 we 2 we place it in MBL, 1 in CMBL, the other one in TCL. So all these 4 machine is well calibrated, maintained for the advanced identification and precise identification of your gene of interest. And in fact, we regularly run a workshop on A to Z PCR, starting from extraction of the RNA or DNA until designing and running the what I call the samples. So Miss Liana's basically a very competent staff and experienced staff which can help you to explain how to use this machine, and in fact you yourself can hands-on this machine. And in fact, I just forgot to inform you on 22nd until 24th, we're going to have another session of flow cytometry workshop. So we still have 4 slots available for you. That workshop will educate you A to Z how to use this flow cytometry in your research, and you will get the certificate, competent certificate. So then once you have the certificate, we will allow you to operate the machine on your own.
PCR results, having a well-calibrated pipette is a key. Let me show you our calibration facility.
All right, now I just want to ask you, how many of you have calibrated your pipette? So pipette is a very important tool for the precision measurement of whatever samples that you are measuring. So make sure that your pipette in a top what I call maintained and a calibrated level. So then you will get a precise what I call measurement of your sample. So we have FOMCAL, what I call pipette calibration unit.
Hi, welcome to Faculty of Medicine Calibration Laboratory of FOMCAL. Are you curious about your pipette and weighing balloon performance? In order to ensure your pipette and weighing balloon are working accurately, this device needs to be checked and calibrated on a regular basis. We provide calibration for pipette and weighing balloon at affordable price.
All right, talking about the calibration again. So Mr. Zul, and there is a few other staffs from the MD lab. So they already have certifications, competent certifications to calibrate your pipette. In fact, we are in the process of getting ISO for this calibration with the collaboration with the industrial partner. And in fact, the cost is much more affordable. So please do send the pipettes to us and get your pipettes calibrated in a regular time.
Mini lecture hall available for researchers' use.
All right, so what you can see here is we have in-house lecture hall which can cater for 50 packs. So basically MBL with the other subunits, we have a complete what do you call, I would say, a package to run a conference workshop. So please come and find us if you want to organize your own seminar workshops. Please do collaborate with us for in fact, we can give a very good pricing. In fact, if you're not charging anything for your seminar or your workshop, and we can consider to give a free of charge for usage of certain facilities under our unit.
The translational core laboratories offer lab services to assist the clinicians with their research needs.
Welcome to our TCL, to our facility.
All right, TCL, as I mentioned, it's based at the 11th floor of the KWKK in one of the UMMC buildings. So this lab is basically provide all the basic tools and facilities for the translational medicine.
This is our inverted fluorescent microscope, which comes with 3 different filters. It also has live imaging capabilities, allowing you to capture real-time cell activity. Here is our cell culture facility. Occupied with biosafety cabinet, inverted fluorescent microscope, CO2 incubator, and also we have offer hypoxia chamber. We offer microplasma testing for cell culture sample using RT-PCR method.
All right, if you're curious whether your cells or your culture got any sort of contamination, especially the mycoplasma or fungal, so we do provide mycoplasma detection service. We use a specific kit to measure, and we provide the what they call the outcome to show whether your culture got the contamination. All right. So as you saw just now, we have a inverted fluorescent microscope where you can do a real-time analysis in terms of proliferation or any specific expression of the surface markers or cellular proteins.
Cell and Molecular Biology Laboratory features cell culture suite and dedicated laboratory space for collaboration.
Hi, welcome to CMBL. Let's have a quick lab tour. Here is our workstation area. We have 24 individual workstations equipped with 2 plugs point, 1 internet port, and a personal locker for your convenience. We also have a lab bench for you to do your experiment.
All right, so as you can see, we have a work suite. So we have a 24 individual work suite station for you to rent out. So and in fact, we have a bench to rent out as well.
CMBL also provides cell culture facility which include CO2 incubator, inverted microscope, BAC for your cell culture experiment.
All right, so as you can see, so we have A to Z equipments or facility to run the cell culture activities. So for the young researchers who got a grant but you haven't got your lab or the benches to start up your research, so please approach us. We can give a very good package for you, to initiate your your research project.
Mana ni ya? Sebelah store lah.
Hi, may I help you?
Hi, from Moderator. I have made an order for pipette tips. Can I collect it?
Okay, wait I check first your order.
Okay.
Okay, yes, I've checked your order. So here is the item that you have ordered. So is there anything else I can help you?
That's all for now. Thank you.
Welcome. So supply store, we provide you with fast and easy access to research lab consumables. We are a one-stop center for plasticware, cell culture media, molecular reagents, and chemical solvents.
All right, so Ms. Munira just introduced the supply store, which located just close to the center point, the open space. So basically, the supply stores has various basic consumables for you to use. And in fact, you come and you can order or make a special order where we collaborate with various vendors to supply a specific consumables if you need it. And we have a specific what they call online booking. So they call research card. However, this system will be replaced with a new system in 2 months' time. However, we will notify you about the new system, how to make an order under the supply store. All right, so this supply store functions like touch and go. So you can top up your balance, and then each time when you purchase a specific consumables, so we will deduct from there. And time to time, Ms. Munira will notify you if, let's say, your funding is getting lesser or need to be top up. Hi, come in. Hi, thank you for joining CRLs. I'm so glad you could join us. Here at CRL, we offer a wide range of facilities and services designed to support your research. From the MBL to the supply store, we have everything you need to conduct cutting-edge experiments in proteomics, genomics, and cell culture. Need help getting started? Our experienced MLTs are always available to provide guidance, training, and support. Whether you're a seasoned researcher or just beginning your journey, we are here to help you. And don't forget about our workshops and events. These opportunities allow you to connect with other researchers, learn new techniques, and stay up to date to the latest advancement in your field. So take advantage of our resources and facilities. We are committed to helping you achieve your research goals. Thank you again for visiting CRL. All right, so we offer various workshops time to time. As you guys can see, the flow cytometry workshop is going to happen very soon, 22nd to 24th, if not mistaken. 4 more slots are available, so please approach us as soon as possible. You can contact Miss Nadia or Miss Tiba for the booking. And also we have qPCR workshop, A to Z workshop with the collaboration with the industrial partner. And also I would like to highlight that we will be having a Western blot, A to Z Western blot technique workshop very soon with the collaboration with the Department of Physiology and the industrial partner. So we will notify regarding that as well. And also upon request, we're going to have kids science laboratory workshop. So the kids science used to be open for outsiders. So now we're going to have this kids science workshop just for the the kids of our staff. So if you want to book the equipment, please do browse Point for the booking. So all the missions are available there. So this time the Kid Science Lab workshop just for our kids. So once we announce, please make a booking and send your kids to enjoy this 2 days event. So if you have any further queries, please do contact us. We have separate emails for the Proteomics, Genomics, and FOMCAL. So send emails, or please do make a call. So we will here to entertain you and facilitate you. Thank you very much. So at this point, I want to express my sincere gratitude to our CRL staff for their dedication and hard work in preparing this presentation in such a short time. We just started to prepare this video last week. And also a special thanks to Mr. Hanafi from YCOM, who really helped us to, to record and editing this video. And this video will be available in a Faculty of Medicine website for you to view. Thank you very much.
Thank you, Dr. Krishna. Hope the presentation has provided everyone with valuable insights into the facilities and services available at CRL for researchers' use. At the moment, we don't have any question from the audience. So it seems that Dr. Krishna's explanation of the facilities and services was both thorough and clear. If any questions arise later, please feel free to email them and we will be happy to address them. With that, we will end today's session. Thank you and have a great day ahead.
Thank you. Bye.